Dear ladies and gentlemen, it's my great honor and pleasure to introduce and moderate the webinar we have today, which is the safety signal detection and analysis for COVID-19 vaccines at the Uppsala Monitoring Center. I think this webinar, before we go to that, first let me say a couple of words of myself. My name is Dr. Lembi Trago, and I am the Secretary General of the Council for International Organizations of Medical Sciences, shortly SIAMS. And it's my pleasure to moderate this webinar. We are living in days of pandemic, and of course, to treat and to get out of it and to have the meaningful outcome of the emergency we have, the vaccines have been absolutely vital tools. And this is why we should also be very much concerned about the vaccine safety. And this seminar is about the safety signal detection and analysis for COVID-19 vaccines at the Uppsala Monitoring Center, which is hosting the global database of individual case safety reports from all over the world, because practically all national pharmacovigilance centers are members of the WHO International Program for Drug Monitoring. Now, the unprecedented time we're living is also characterized that we have more than ever individual case safety reports reported to the global database. And the vaccines, COVID-19 vaccine-related individual case safety reports are likely already today in Uppsala Monitoring Center databases counting more than 1.2 million. And of course, this massive influx of individual case safety reports about the COVID-19 vaccines is posing new opportunities and also new challenges. But let's get to the business, and it's my pleasure to introduce the first speaker. The first speaker we have is Ms. Helena Sköld, who is the Vigibase manager and has been working in Uppsala Monitoring Center since 2007. And she has formerly been also working for the Swedish Regulatory Authority, the Medical Products Agency, and she is today responsible to the data in Vigibase and how it is used. So, Helena, please, the floor is yours.
Hi, my name is Helena Sköld. I'm Vigibase manager at the Uppsala Monitoring Center. And I will be telling you a little bit of background about the WHO's Program for International Drug Monitoring and the global safety database Vigibase, with a bit of focus on the COVID-19 vaccines. I'd like to start us off with a short reminder about what pharmacovigilance is all about. Back in the 1960s, Dr. McBride made an observation of rare malformations in newborns born to mothers that had taken thalidomide. He compared this to what he would have expected in terms of frequency, and he noticed that this happened unexpectedly often. He then asked the vital question, did anyone else see this? This unfortunate and tragic story about thalidomide was the background story for how the WHO Program for International Drug Monitoring came into existence, and this is why we have Vigibase today. Vigibase is there to help us answer that question, did anyone else see this? And it lets us find the rare and unexpected. In order to prevent future tragedies of this kind, WHO, the World Health Organization, set up the Program for International Drug Monitoring with 10 pilot countries in 1968. Today, 2021, this program have 148 full members that are sharing their safety data for drugs and vaccines, and 23 associate members that would like to do the same, that has expressed this wish. Together, these members, they cover over 98%, almost 99% of the world population, and the database, VigiBase, contain in total almost 28 million case reports. Of these, about 1.8 million cases originates from the COVID-19 vaccines from over 120 countries who has already shared this data. The support for the WHO program and its operational activities were transferred to the Foundation WHO Collaborating Center for International Drug Monitoring set up in Uppsala in 1978. It's a mouthful to say, so today we're known as Uppsala Monitoring Center, or UMC for short. And we provide the scientific, technical and operational support to the WHO program. UMC are also the custodians of VigiBase, and we are a standards development organization for WHO Drug Global, which is a global drug reference used for many years as a de facto standard in clinical trials, increasingly now also in the post-marketing area. In this SDO role, we are also actively involved in supporting implementation of the ISO standards for identification of medicinal products, the IDMP. UMC also provide IT solutions aimed to strengthen the national pharmacovigilance systems of the WHO program members, which most commonly would be the National Regulatory Authority, NRA. And we are also actively involved in global harmonization efforts of the International Standards Organization, ISO, and ICH to support safer use of medicines. Let's talk a little bit more about VigiBase and explain some basic concepts. The WHO program members, usually the National Regulatory Authority, as I mentioned, they would be responsible for the safety of products in their markets, and they would collect ADRs and AEFIs, adverse drug reactions and adverse events following immunization, from patients, physicians, industry, and other stakeholders within their national systems. Now, VigiBase collect the data from the program members and only from them. So the other stakeholders always need to report to their national authority. VigiBase today holds, as I said, 28 million case reports from 148 members. If we look at this data, almost half actually originates from the US and about 25% from EU and UK. Many of these members, they have been reporting and shared their data to VigiBase for over 40 and 50 years for the entire length of the program. Asia, however, is very rapidly increasing their shares, and also the proportion of data originating from low and middle income countries, they have increased their share over the past 5 to 10 years from 5% to almost 15% today. And this really makes for a uniquely diverse and comprehensive data source. Now, the IT solutions that I mentioned in the previous slide, they support the program members both in data collection, data management, and analysis in their national setting to support their mission for safe products in their market. VigiFlow, for example, this is a data collection and management system used by over 90 members as a national safety database. And in Vigilize, they have a powerful analysis tool free of charge that can analyze national data as well as data in regional collaborations with instant access to VigiBase and what others have already experienced as a reference. Signals found by UMC and other members are also available within Vigilize. At the UMC, VigiBase data is used to complement national safety analysis with statistical signal detection on a global level, and also for method development to handle this amount of data and to support the identification of safety signals, which you'll hear more about in my colleagues' presentations. A very important aspect of VigiBase is the structure and coding applied. With this many different data sources, it's really necessary to have a common data model and to ensure that the data that is to be used for analysis is also appropriately structured. In VigiBase and in all our IT solutions, we use WhoDrug to code the drug information and MEDRA, the Medical Dictionary for Regulatory Activities, to code reactions, events, tests, and indication. If the information isn't properly structured, it doesn't really exist, statistically speaking. It's really hard to find. And even though we do have this common data model and we have appropriate coding, it's still important to remember that these many different sources also mean that the data is very heterogenic, as countries may have their own national legislations, guidelines, and policies to follow on what they can and cannot share. Other stakeholders can also access Vigibase data, and this would be with a bit of limited level of detail. Now we'll get back to this in my last slide. Now moving on to the current pandemic and some of the challenges with the global vaccinations in particular. With the ongoing rollout of vaccines, including new innovative technology on a massive scale globally, fast safety data exchange is essential. So if we consider that when millions receive their vaccinations each week globally, even very rare events affecting only maybe 1 in 100,000 or 1 in a million can potentially affect hundreds or thousands of vaccinees in only 1 week. With this perspective, it's obvious that efficient data collection and sharing is crucial. Learning from each other's experience as early as possible is important to support the global vaccination effort, and this makes Vigibase more important than ever before. This is why the manual that WHO issued, the COVID-19 Vaccines Safety Surveillance Manual, recommends countries to review and adapt processes for timely reporting, reviewing, and data sharing nationally, regionally, and globally. Since many countries today still, to a large extent, actually have based their reporting systems on paper, this is a major challenge. It's not uncommon that it takes a paper form up to 6 months from when it was created until it's received by the regulatory authority. As I'm sure most of you recognize, our entire operations at the UMC were affected by the pandemic. We needed to ensure that the members of the WHO program could meet the challenges of a large-scale COVID-19 vaccine rollout. So we wanted to help them follow the WHO guidance, and we completely disrupted our plans to focus on responding to the pandemic situation in a number of different ways, affecting all of our activities. And I will list some examples for you here, and some of these examples will be covered by my colleagues' presentations. One of the activities was to create a descriptive report of the COVID-19 vaccine reporting. And before the vaccines came out, this was covering the treatments for COVID-19. Our signal detection has been focused on the COVID-19 vaccines, and we utilize our methodological development and our research, like cluster analysis, for example, on the COVID-19 vaccine surveillance. We also participate in expert groups in the COVID-19 vaccine context. And with the global drug reference that I mentioned, WHO Drug Global, we ensure that there is coverage of all different COVID-19 vaccines in order to support identification globally and to enable effective and appropriate analysis. This is not a straightforward task, as countries actually have their own ways of naming these vaccines, depending on the regulatory status and other factors. For example, the Pfizer vaccine, Comirnaty, used in Europe, has at least 4 other names in other parts of the world. It was recently now approved by FDA as Comirnaty as well, which kind of facilitates things. In the IT solutions we provide to our WHO program members that I mentioned, Vigiflow, Vigilize, we've made a lot of vaccine-focused development to improve usability in collecting and analyzing vaccine safety data. This was done in close collaboration with WHO headquarters to ensure that we also meet the needs of the national immunization programs that are oftentimes not under the same organization as the Drug Regulatory Authority. To ensure access to qualitative and consistent data for analysis, we also put some manual efforts into the specificity of coding in the incoming vaccine data. So, can anyone access Vigibase? I promised to get back to that on my last slide, which this is. So, there are a few different options. VigiAccess is updated twice per week. It's available as a website, but also as an API service. This is a public data set, but it only contains counts and no case level data. So basically it provides a statistical representation of the data in Vigibase with some important limitations, like we're grouping data by continent, by substance and by age group. And in particular, this is important to know if you search for a specific trade name, the result will always be presented for the substance. And in the COVID-19 vaccine context, this means that all of the COVID-19 vaccines are actually represented as one substance, and this means that it's not possible to present statistics per specific vaccine in VigiAccess today. Another option is our customized search service. This is where we have some expertise here that can help you define the search criteria and also to tailor the output to what you need to have. This dataset is also updated twice weekly. And the last option on the slide is the Vigibase extract case level. This is something that we provide for vendor tools that use the data as, for example, for background in signal detection, statistical signal detection. This dataset also has some limitation as with the other datasets. So this is all based on what can be shared publicly for Vigibase data at this point. All these services, except for the vigiaccess.org website, comes to cost, which is what we need in order to have cost coverage for the structuring, coding, and processing taking place in order for us to be able to provide this data. There's also an evaluation process in place to ensure that potential data recipients that aim to use the data have their purposes in line with the purpose for which it was collected, which means for safer patients and for safer use of medicines and vaccines. Now I've come to the end of my presentation, so I thank you very much for your attention, and I hope that I've shed some light on who we are and what we do and the important role that Vigibase has to play in the COVID-19 context. Thank you.
Thank you, Helena, for your excellent presentation. And of course, I would like to mention now that we take the questions in the end. In the end, we have panel discussion, and we have already a number of questions received for this. And of course, I see that also in chat there are new questions coming. So please be patient. We try to address as many as possible your questions during the panel discussion in the end, where some additional people to the speakers will be joining the discussion. But now let's go to the next presentation, which is descriptive analysis of COVID-19 vaccines reporting in Vigibase. And this presentation will be given by Ms. Sarah Widlin, who is data scientist and has been working in research at UMC since 2015. She works with programming and software prototyping, data extraction, computation and analyzation, method and algorithm development research. And she is, since the pandemic, working very intensively focusing on the pandemic-related issues, like presenting statistics from Vigibase regarding COVID-19 treatments, and since 2021 also about the vaccines. So Sarah, please go ahead with your presentation.
Hi, my name is Sarah Widlin and I work as a data scientist at Uppsala Monitoring Center. I will present a descriptive analysis of COVID-19 vaccine reporting in Vigibase. This is the joint effort of the persons listed on this first slide. The background is that UMC produces descriptive overview reports monthly, describing the COVID-19 vaccine ADR reporting in Vigibase. These descriptive reports are available through UMC's pharmacovigilance tool Vigilize if you are working at the National Center within the Program of International Drug Monitoring. That is also where all the data comes from. It has been shared from national centers throughout the world within the context of this program. Today, we will take a look at the August report. Let's dive in. The intended use of the descriptive report is to provide an overview of the COVID-19 vaccine ADR reporting and provide supporting information to staff at national centers. I'd like to stress that the usual caveats for spontaneous reporting applies. No manual assessments has been performed and the intended use is a descriptive overview. Let's start by looking at the vaccine products reported into Vigibase. We have, up until today, in total a bit over 1.6 million COVID-19 vaccine ADR reports. The majority concerns the Pfizer-BioNTech vaccine, followed by the AstraZeneca vaccine. Vaccine products with less than 100 ADR reports are grouped together in an other category, and ADR reports without information about vaccine products administered are placed in unspecified. Here we look at the reaction onset dates. The graph tries to illustrate reporting delay. The red ADR reports are shared into Vigibase since last compilation, so during the summer. The majority concerns ADR occurring during that period of time or the month before, but still many are describing events occurring all the way back to January. So we can see that there are some lag times before we get the ADR reports into Vigibase. This graph shows the same data, but divided between the different vaccine products. We can see, for example, a peak of AstraZeneca around March, and at the same time an introduction of CoronaVac and the Johnson vaccine. Countries that have shared the largest amount of ADR reports are the US, UK, Netherlands, Italy, France, and the Philippines. Pfizer-BioNTech originates from a variety of countries, but 40% of the ADR reports comes from the US. And a similar situation with the AstraZeneca vaccine, but here 45% comes from the UK. For Moderna, 75% of the ADR reports comes from the US. And for Johnson vaccine, 71%. CoronaVac originates from the Philippines in all 80% of the cases. And in total, 91% of the Sinopharm ADR reports are shared from Peru, Morocco, and Pakistan together. This is very important to keep in mind when comparing differences between ADR reports of different vaccines, because the patterns we see may be confounded by the main reporting country. For example, so far during 2021, about 30% of all ADR reports in Vigibase were reported as serious. But for ADR reports from the UK, more than 50% were reported as serious. This is driving up the serious proportion for AstraZeneca, that is mainly reported from the UK. This kind of confounding is crucial to keep in mind when comparing ADR reporting patterns of different vaccine types. Another aspect to consider when looking at this data is the possibility of differences in sex and age of the patients receiving the vaccines and reporting the ADRs. The majority, 70% of all COVID-19 vaccine ADR reports consider females. This pattern is fairly consistent between the different vaccine types, except for Covidensia. But we also have substantially less data for this vaccine. Looking at the age box plots to the right, the boxes represent the interquartile range, so the range between the 25th and the 75th percentile. and the vertical bars inside the boxes shows the median value. We can see that the median age for all vaccines is approximately 50 and ranges between 35 and 55 years for the various vaccine products. We will now shift focus a bit and look more on the outcomes of the ADR reports, so the suspected and reported adverse events. The first question would be where to direct our attention. You may or may not be familiar with MEDRA, the Medical Dictionary for Regulatory Activities. This is a hierarchical terminology containing thousands of terms you could use to describe medical events. A suggestion where to start comes from the Brighton collaboration. The Brighton collaboration, in partnership with Coalition for Epidemic Preparedness Innovations, have described adverse events of special interest, AESIS, as high priority adverse events for monitoring after vaccination. And UMC has mapped a version of the AESIS to MEDRA. So for example, an ADR report would contain the AESI cardiac failure if it contains any of around 100 MEDRA terms, including cardiac failure acute, myocardial depression, pulmonary congestion, and so on. Some AESIs are also more complex and have restrictions on age or require reactions from more than one set of terms. In the leftmost panel on this slide, we can see the proportion of adverse events of special interest reports for the different vaccine products. It's between 10 and 15% AESI reports for the 4 big vaccine products, and generally lower for the ones where we have less data. On the opposite side, the rightmost panel displays the proportion of fatal reports for all vaccine products within a few percent. As a comparison, it's good to know that the number of fatal ADR reports from this year in all of Vigibase is 3%. Now lastly, in the middle panel we look at serious ADR reports. It is possible to flag the ADR report as serious. And as we can see, 23% of all COVID-19 vaccine ADR reports are flagged with serious. As a comparison, 32% of all ADR reports in Vigibase from this year have this flag. The proportion for AstraZeneca is higher. But as I previously mentioned, that is expected since almost half of the AstraZeneca ADR reports comes from the UK, and the UK generally reports a higher proportion of serious. Question is though, is the increased serious proportion compared to the other vaccine products only due to confounding by country? Or can we see a real effect due to the vaccine itself? And how to know? Let's revisit the origin of the AstraZeneca ADR reports. The 8 countries contributing with the greatest number of AstraZeneca ADR reports together stands for 78% of all AstraZeneca reports in Vigibase. If we look at the serious proportion for AstraZeneca from these 8 countries only, we get an even higher proportion, 40% serious. And if we also look at the serious proportion for Pfizer-BioNTech for these countries, and then calculate what the serious proportion would be for Pfizer if the ADR reports would come from the same distribution as the AstraZeneca ones from these countries, we can calculate the adjusted Pfizer-BioNTech serious proportion to 30%. So in conclusion, even when we adjust for reporting country, it seems like the AstraZeneca ADR reports are reported more often with the flag series than Pfizer-BioNTech. So in this case, the noted higher proportion serious ADR reports might not only be confounded, at least not only confounded by country. Now, let's drill down into the actual AESIs for some of the vaccine types. Here we see Pfizer-BioNTech. The different AESIs is the y-axis, the percent of ADR reports is the x-axis, and the colors represent different age groups. The top 5 ASs are hemorrhagic disease, anaphylaxis, cardiac failure, anosmia ugicia, and cardiac arrhythmias. And if we compare the graphs for the 6 vaccine products with the greatest number of ADR reports, we will actually see that 4 of these ASs are in all top 5 lists across the vaccine products. AstraZeneca here has the same top 5 except for cardiac arrhythmias, and instead has deep vein thrombosis. Moderna has the same top 5 as Pfizer-BioNTech, but in a different order. And the Johnson vaccine has the same top 5 as AstraZeneca, but in a different order. CoronaVac shares 4 ASs with the rest, but also throws in vaccine-associated enhanced disease. And lastly, Sinopharm shares 4 ASs with the rest, but also includes ischemic heart disease. So, 4 of these ASs seem to be the most commonly reported across all these 6 vaccine products. That is anaphylaxis, anosmia ugicia, cardiac failure, and hemorrhagic disease. Now, let's take a step back, drop the ASs, and simply look at the reported terms. Here we use MedDRA preferred terms, PTs. These are the most commonly reported terms for all COVID-19 vaccine ADR reports. Headache, pyrexia, fatigue, chills, myalgia, nausea, arthralgia, malaise, dizziness, and injection site pain. And drilling further down, we can divide by vaccine product. Here are the same top 10 terms, but divided by product. To the right, you can also see the union of all top 10 terms for each individual vaccine product. This way, we can compare and see that, for example, headache is more commonly reported for AstraZeneca than for Pfizer-BioNTech, and that, for example, injection site pain is much more commonly reported for Covaxin than for any other vaccine product. The purpose of this presentation was to show you our monthly descriptive overview of the COVID-19 vaccines, in order to give you an overview of the COVID-19 vaccine ADR reporting in VigiBase. This can serve as a background for further reasoning and in-depth manual assessments. Using a global database enables rapid detection of emerging safety concerns, but as we also seen, it comes with additional complexities, for example, confounding by reporting country. Thank you for listening. I hope you were inspired by this presentation. If you have access to Visualize, make sure to log in and dive deeper into this report, or make your own searches for what you are interested in.
Thank you, Sarah, for your interesting and excellent presentation. And as said, we take the questions in the end. So now it's time to move on to the next presentation, clustering prospectively to find multi-term adverse events in the COVID-19 vaccines. And this presentation is given by Dr. Jim Parrett, who is also data scientist from the Uppsala Monitoring Center. And he has 7 years experience in analyzing data. He holds also PhD from the University of Birmingham, UK, where he focused on advanced techniques for the data analysis. So he has worked also 2 years as a software engineer in the Artificial Intelligence Research Department at Klarna, and he joined the Uppsala Monitoring Center in March 2020. And I think he's right in the middle of developing novel methods for signal detection. So please, Jim, go ahead.
Hello everyone, and welcome to this presentation about some of the work being conducted at the Uppsala Monitoring Center in the effort to monitor the safety of the COVID-19 vaccines. My name is Jim Barrett, and I'm a data scientist with one of the research groups here. I'll be talking about how we're using clustering algorithms to look for patterns of adverse events characterized by multiple terms. Here is the agenda for my talk today. I will start by describing the problem we're trying to solve. I'll then give a high-level overview of the tool we've chosen to tackle this problem, namely clustering. And then I'll describe how we're using it to monitor the safety of the COVID vaccines. And finally, I'll give a specific example of the kinds of things we find. How do we most effectively search through large numbers of adverse event reports to identify plausible signals? This is the key question at the heart of a large fraction of the work we do at the UMC. Bear in mind that for some medications, we have hundreds of thousands of adverse event reports in Vigibase, which is clearly far too many to assess by hand. Naturally, this is a problem that has existed in drug safety monitoring for as long as large databases of adverse event reports have existed, and as such, there have existed approaches for prioritizing and sorting them. The most common approach to finding potential signals is to ask the question, are we seeing this adverse event more often in combination with a drug than we would generally expect to see it occurring? Using the data in Vigibase, we can compute a good estimate of how often we might expect to see an individual adverse event. And by comparing this to the number of times we observe that event in combination with the drug in question, we can immediately see which adverse events are happening more often than we expect. Say, for example, that we have 10 million reports in Vigibase. And of those 10 million, 10,000 of them have a certain adverse event, which is 0.1%. If we then say that we have 20,000 reports from a certain drug, and we see that 400 of those reports have that same adverse reaction, which is a 2%, this is a higher rate than we expect, and this drug adverse event combination clearly demands looking into. This approach is known as disproportionality analysis. It is used routinely at the UMC and is very successful in highlighting potential safety concerns for further investigation. However, consider that for some drug, we have a case series of 3 reports. The first report has the 3 adverse events, nausea, migraine, and vision abnormal. The second report has vomiting, migraine, aura, and photophobia. And the third report has vomiting, headache, and aura. These 3 reports seem to clearly describe the same type of event. The patients have all suffered migraines. However, notice that whilst all 3 reports do use similar terms to describe what has happened, there is no one term which appears on all 3 reports. If we were to perform traditional disproportionality analysis, we wouldn't find all of these reports, since disproportionality analysis only deals in one term at a time. If we want to be able to group these reports together, we're going to need a method which looks beyond individual terms. So, to summarize what we're looking for here, we're asking, can we automatically identify clinically coherent groups of adverse event reports? Now, there is some hidden complexity in the way we have phrased this question, namely that we are interested in clinically coherent groups of reports. This may seem like a no-brainer requirement, and it is, but it's worth highlighting here because there exist many methods which could group together reports in a way which makes no clinical sense. Any method we choose should be optimized towards making the groups of conditions useful for signal detection and for signal analysis. Now, I'm going to make a small terminology change for the rest of this presentation. Whenever a data scientist hears people talking about automatically finding groups of things, there's one word that springs to mind, which is clustering. In the context of what we're discussing for the rest of this presentation, the word cluster is just a synonym for group. So, can we automatically identify clinically coherent clusters of adverse event reports? There is a whole family of data science algorithms known as clustering algorithms, which are designed to solve problems exactly like this one. I'll spend a couple of minutes now giving a very high-level introduction into what they are and how they work, and then we'll talk about how we're using them. Fundamentally, clustering algorithms serve to find patterns in data by identifying which data points are similar to one another. The data points which are similar to one another are automatically assigned to the same cluster by the algorithm. If you look at this simple picture of an illustrative dataset, You may already be able to see with your naked eye a couple of groups of data points which are more similar to one another compared to the randomly scattered background. Finding clusters of data points like this, automatically, amongst hundreds of thousands of data points, is exactly the remit of clustering algorithms. Of course, you might reasonably be saying to yourself right now, sure, Jim, but there's a long way to go between dots scattered neatly on a two-dimensional graph and lists of adverse events on an adverse event report. And that's a fair point. And of course, our problem is much more complex, but conceptually, this picture is a good one to keep in the back of your mind when we're talking about clustering. Now, I don't want to go into too much of the gory details about the specifics of the clustering algorithm we've developed at the UMC. However, I think it's useful to outline the core assumptions we use to drive the clustering algorithm. The first assumption is that each report we receive is talking about just one clinical condition. The second assumption we make is that each clinical condition has some probability of occurring in a population of people who have taken a drug. And the third assumption is that given that a patient has one of these clinical conditions, that they have a certain probability of experiencing each symptom or adverse event. Collectively, these assumptions define what we call a probabilistic model, and there exist many mathematical tools for fitting them to data. The specific clustering algorithm developed at the UMC has been under development for many years, and I want to give a brief illustration of the types of clusters that it highlights when we run it on a data set. Risperidone is an antipsychotic medication, and its set of adverse event reports in Vigibase was used as one of the first test cases when we were developing the clustering algorithm. And I'm going to show you 3 illustrative clusters that appear from running the algorithm on these reports. First, we have this cluster. There are 1,883 reports in the cluster, with an average of 3.2 adverse events per report. The most commonly reported adverse events in the cluster are on the left, and their reporting frequency within the cluster is on the right. This cluster is a clinically coherent picture of a worsening of the patient's underlying condition. The algorithm also found this cluster, describing a spectrum of extrapyramidal disorders. And it also found this third cluster, describing disorders related to the female endocrine system. What I wanted to demonstrate here is that this method appears to have come a long way towards achieving our aim of finding clinically coherent clusters of adverse event reports amongst a data set of adverse event reports. I also wanted to briefly reiterate the key difference between using clustering to find signals compared to using the disproportionality analysis techniques I described at the beginning of my presentation. Disproportionality analysis works on the level of individual reported adverse events, whereas clustering tries to find patterns by looking at the adverse event reports, collections of adverse events. These are fundamentally different approaches, and we don't expect one to be universally superior over the other. So, how are we employing the clustering algorithms in our efforts to monitor the safety of the COVID-19 vaccines? I hope I've managed to convince you so far in this presentation that clustering is a promising approach to signal detection. However, the approach is still in its infancy. So far, all of the studies we've conducted with this clustering algorithm have been retrospective. That is to say, we have had a large and stable population of adverse event reports received over a long period of time. This is certainly not the case for the COVID vaccines. We are using the COVID vaccines as an opportunity to build our knowledge and capacity for using the clustering algorithm prospectively to look for potential safety concerns as they occur. So, more concretely then, what are we doing when it comes to clustering and the COVID vaccines at the UMC? Each week, we receive a new batch of adverse event reports into Vigibase, which typically total a few tens of thousands of new reports for the COVID vaccines collectively. As of the time I'm recording this presentation, which is the 23rd of August 2021, there are approximately 1.8 million COVID vaccine reports in total in Vigibase. We've been regularly clustering the COVID vaccine 19 reports since February this year. For most of the year, we've been running the algorithm every week. However, for most of the vaccines, the view that clustering gives us has become very stable, and so we've recently reduced this frequency to about once every 3 weeks. We cluster the reports for each vaccine independently. A constraint of the clustering algorithm is that we require a minimum of around 1,000 reports for it to work effectively. And the vaccines included in this constraint right now are the Pfizer, AstraZeneca, Moderna, Janssen, Sinopharm, Sinovac, and Sputnik V vaccines. We've also been running the algorithm on the reports of vaccines grouped by their platform. After each application of the algorithm, team members with clinical expertise perform a review of the clusters, paying close attention to clusters which are clinically unexpected, clusters which have grown since the previous time we've run the clustering algorithm, and clusters which are new. Some clusters are then marked for additional in-depth analysis. The team then works together to improve this process and to come up with new ideas for how we can make the clustering better for the next time. For the larger vaccines, we can see hundreds of clusters each time we run the algorithm, all of which need to be reviewed by experts. Each cluster can have tens, hundreds, or even thousands of reports in them. Since, as I mentioned on previous slides, we are rerunning the algorithm on several vaccines and groups of vaccines regularly, the amount of information for our clinical experts to review quickly becomes very large. This makes the presentation and accessibility of all this data very important in order to make reviewing the data as efficiently as possible. In order to address this challenge, we've developed an internally accessible website which is automatically populated each time we run the clustering algorithm with all of the clusters, all of the vaccines, and all of the vaccines grouped by platform. And for each cluster, for each vaccine, and for each application of the clustering algorithm, we give a broad summary of the clinically pertinent information, which looks something like this. Here I'm showing one of the largest clusters we found in the reports for the Pfizer vaccine, up to the beginning of August this year. I'll zoom in for a closer look at the information we display now, looking at the bottom half first. The bottom half of the summary gives an overview of the specific adverse events associated with the cluster. Here we show those adverse events which are reported in at least 10% of the 33,000 reports in this cluster, and how many of those reports has each of these events. This kind of information allows our pharmacovigilance experts to quickly assess the clinical picture which is represented by this cluster. We also display the time to onset of each of these reported reactions. And these links take the user to Visualize, which Helena mentioned earlier in her presentation, which allows the clinical experts to see additional information about the reports, including the free text case narratives where they exist. The top half of the cluster summary shows other information that's useful for the clinical experts both to understand the clinical picture behind the cluster and to get an overall picture of the severity of the reactions the patients are having. We show the total number of reports in the cluster and how many of those are new since the previous application of the clustering algorithm. We present a summary of demographic information, such as the gender and age distribution of the patients. And we also display various counts related to how many of the reports in the cluster match criteria for adverse events of special interest, what fraction of the reports in the cluster have been reported as fatal, serious, or as important medical events. Armed with all of this information at a glance, we're able to make rapid assessments of each cluster generated by the algorithm, and then prioritize what is most interesting to follow up on with more detailed analysis. I'd like to finish by giving a concrete example of the clustering algorithm finding a previously unknown reaction among the COVID vaccines. On the 5th of August this year, the European Medical Agency's PRAC committee updated the product information for the Janssen vaccine to include the adverse event tinnitus, based on 6 cases observed in clinical trials and 108 further cases observed in post-marketing surveillance. The British Tinnitus Society also described tinnitus as a rare side effect to COVID-19 vaccinations. Running the clustering algorithm on the Janssen vaccine, we see the following cluster. Here we display the reported adverse event on the left and the fraction of reports in the cluster reporting each event on the right. This signal was also highlighted with classical disproportionality analysis. However, the fact that it was also highlighted by cluster analysis increased confidence that the statistical pattern was real and was a valuable tool for finding potential additional reports for the case series provided to our clinical experts for analysis. So, to conclude my presentation, clustering is a fundamentally different approach to finding clinical patterns in adverse event reports. We're using this approach prospectively at the UMC to identify and investigate potential signals, and the method has been incredibly promising so far for discovering signals amongst adverse event reports. Thank you for listening to my presentation, and I look forward to answering any questions you might have.
Thank you, Jim, for this excellent and inspiring presentation you had. We are now moving to the next presentation, the last presentation before we go to the panel discussion. And the last presentation is detecting potential safety signals regarding the novel COVID-19 vaccines on a global level. And this presentation will be given by Ms. Annette Rudolph. Annette is a pharmacist from Germany, from Bonn. And actually, she became acquainted to the pharmacovigilance when she was on the traineeship at the University Hospital in Zurich in Switzerland. After that, he had also an experience working 1 year in Italy in Veneto Regional Pharmacovigilance Center. And then in 2020, she joined UMC and is currently focusing on various signal detection activities. So Annette, please go ahead. Thank you.
Hi, my name is Annette, and I am working in the team focusing on signal detection here at UMC. In the upcoming 15 minutes, I will take you through our signal detection activities for the COVID-19 vaccines. Just briefly to recall what we are dealing with. A signal is essentially a hypothesis of a risk of a medicine, supported by data and arguments that can be derived from one or several possible sources. So the first question I want to address is, why is UMC pursuing signal detection activities? International regulators such as the US Food and Drug Administration or the European Medicines Agency, as well as national authorities, have drug and vaccine safety surveillance systems in place and perform signal detection. Our position as managers of VigiBase allows us to have a look on the bigger pictures from a bird's-eye perspective and concentrate on safety issues that might not be apparent on a smaller national scale. So our aim is not to repeat what others are doing, but rather to complement activities performed on the national and international level by maintaining the global perspective. Furthermore, our position allows us to focus on process improvement and method development. Now let me briefly take you through our signal detection process. Reports on suspected adverse drug reactions are collected and processed at national pharmacovigilance centers of the member countries. From there, they are shared into the global database VigiBase. We regularly screen VigiBase for potential safety signal. Due to its size, the first step in our signal detection process is a statistical screening and disproportionality analysis using a tool called VigiRank that I will introduce you to in the next slide. Substance reaction combinations that are revealed during the screening next undergo an initial manual assessment by PV experts. The initial assessment consists of checking whether the adverse effect is already adequately covered in the product information and a review of the case series to exclude other more likely causes. If a combination is found to be eligible for further investigation, then the next step follows in-depth manual assessment. Herein, VigiBase case reports are individually assessed and summarized, and the scientific literature is reviewed for additional evidence. In case it is decided to be a signal, it will be then shared with the member countries via Vigilize and with the broader public via the WHO pharmaceutical newsletter. To come back to our statistical screening, I'll briefly elaborate what VigiRank is. It is a predictive model that ranks drug reaction combinations according to multiple aspects of strength of evidence. Parameters that make up the VigiRank are disproportionate reporting, recent reporting, geographic spread, informative reports as indicated by VigiGrade, and narratives. VigiRank has been successfully implemented in the UMC's signal detection since 2014. Another tool we use is VigiGrade, a multidimensional measure for the completeness and informational content of reports in VigiBase. Information taken into account by VigiGrade are, for example, patients' characteristics, such as age and sex, information on the suspected drug, such as dose and indication, and information on the suspected reported ADR, such as time to onset and outcome. A score then is given to each field if it has information. This is done for every drug ADR combination in a single report. The individual field scores are weighted and combined into a score for the whole ICSR, and in case of several reported reactions, an average is calculated. The maximum possible score is 1.0. When it comes to the manual in-depth signal assessment, there are differences between assessing single case reports and a case series. When assessing single case reports, we apply the WHO-UMC causality categories. Criteria taken into account during causality assessment are the timely association between substance intake and onset of the reported reaction, the presence or absence of alternative causes, information on a positive or negative D-challenge, the pharmacology of the suspected substance in connection with the phenomenology of the reported reaction, and finally, information on a positive or negative re-challenge if available. When assessing a case series for causality, the aforementioned criteria do play a role. But furthermore, the Bradford-Hill criteria are taken into consideration. The two maybe most important criteria for us at UMC are the strength of the association, as measured by disproportionality, and the consistency characterized by multinational reporting. Moreover, temporal relationship, biological plausibility, coherence, specificity, evidence, and analogy are being considered. Following, I'll elaborate a bit more detailed what we did so far regarding the signal detection for the novel COVID-19 vaccines. The COVID-19 pandemic, as well as the fast development of the vaccines, represent an unprecedented situation. So our main scope was to make sure that the detection and assessing of potential safety signals happened in a timely manner. We encountered several challenges during our preparation. The safety surveillance and signal detection for the new vaccines poses a new and unprecedented situation for us. Reasons are, for example, the amount of reports we received in a relatively short period of time, resulting in way bigger case series to assess than we were used to before. Furthermore, the quality of information we receive in the reports is highly variable from country to country. An additional problem we encountered was that the algorithm VigiMatch that we usually use for detecting duplicate reports is not applicable for vaccine reports due to their characteristics, especially during mass vaccination campaigns. For example, patient information, suspected vaccines, date of administration, and symptom onset might be similar in many reports. Despite the short preparation period, thanks to Vigilize, we were able to start with our signal detection activities right away as soon as the first reports were arriving. Criteria we used for the identification of combinations representing potential safety issues were disproportionate reporting, meaning the number of observed reports exceeding the number of reports one would expect based on the calculations run by VigiRank. The seriousness, meaning the proportion of serious reports in a case series, and multinational reporting. Using Visualize for signal detection offers several advantages. The database is frequently updated, making real-time disproportionality analysis and signal detection possible. Thanks to our colleagues at WHODRAC, it is now possible to perform disproportionality analysis based on the individual vaccines, enabling, for example, platform analysis. Visualize includes a nice data visualization surface, facilitating the identification of pinpoint characteristics of the case series. Additionally, it offers features for easy internal communication and follow-up within the organization, resulting in the possibility for everyone in UMC to support our signal detection efforts. If one should seek for disadvantages, I could mention its low plasticity. It is not possible to apply several filters at once, which complicates the prioritization of combinations. Furthermore, VigiRank by default uses the entire database as background for disproportionality analysis, posing the potential risk to distort some combinations due to the high background rates in the database. One example would be rhabdomyolysis caused by statins. In order to obtain more flexibility, we performed some screenings using Vigimine, an internal tool for tailored signal detection, enabling the ordering of combinations lists based on the parameters calculated by VigiRank, as well as the implementation of any metric that might be interesting to include in the disproportionality analysis. One such metric could be, for example, the proportion of reports per number of countries they derive from. This could help detecting reactions that get reported in small numbers, but from various different countries. Such reactions might not get detected on a small national scale, but only when looking at it from a bird's perspective. To conclude, with the last slides, I would like to give you an overview of what else we have planned for the coming month. As mentioned before, our disproportionality analysis is by default performed against the background of all Vigibase. This is in line with empirical findings that the best way to perform vaccine signal detection is to use the full dataset as comparator in order to not miss important information. However, having typical vaccine-related reactions in mind, we will additionally attempt to perform disproportionality analysis using several selected vaccine backgrounds. Considering the vaccinee's population characteristics, vaccines such as influenza and H1N1 seem to be appropriate candidates. Comparing COVID-19 vaccine-related reactions to a complete vaccine background, including typical childhood vaccines, seems to be worth a try as well. With the vaccine rollout proceeding and the approval of the vaccines extending, we will want to focus more on reports from regions with high proportions of low and middle income countries who might have limited resources to perform signal detections themselves. Also, we will want to focus on different patient groups, such as pediatric patients. Due to the large numbers of reports and observed combinations, we are furthermore working on improving our signal prioritization strategy. Criteria we are considering are the global spread of reports, the potential impact of a combination on the public health and patients' quality of life, the already available information from clinical studies and scientific literature, the clinical importance of the observed reaction, as well as the kind of message we could send by publishing a signal. Recently, we started implementing also qualitative signal detection by regularly reviewing the published literature as a complementary activity to our regular database screenings to follow early hints when statistics don't apply. Thanks a lot for your attention, and I will now be happy to answer any of your questions.
Thank you very much for your excellent presentation. And we are now going to the panel discussion where we try to answer at least some of the questions because we have received quite a huge number of questions. We received some questions before the webinar, and now in the course of webinar we have of course received additional questions. Now, before going to the panel discussion and trying to answer at least some of the questions, I would like to also introduce Dr. Gunying Yu, who is a medical director and senior pharmacovigilance expert from Uppsala Monitoring Center. She is a medical doctor and specialized in clinical pharmacology. She is also, of course, having a PhD degree, and she is has been working also with the Swedish regulatory agency, Swedish Medical Products Agency, and while she was working in the MPA, she was also several years member of the European Pharmacovigilance Risk Assessment Committee, the committee which is attached to the European Medicines Agency and actually the major pharmacovigilance decision-making body in a way in the EU context. So happy to have her on board, and now we are starting to have some discussion and questions. And just to kick the discussion going, I have one question. Let's propose that you in the UMC, you find a signal, and then you communicate it to the let's say the US FDA or the Europeans who do their own signal detection in their databases, and then you find out that what you have found out is not exactly what they have found out, and what you see it as a bit of problem, they may not see it as a problem. So, and then let's imagine that somebody has done also some signal detection on the country level, which is smaller, and then how much, and there are discrepancies between the signal detecting activities between different environments, and how you explain these discrepancies, or if they are not there, then of course this question is valid. So who wants to respond to that?
I just would like to start out by saying that what the UMC provides to the National Center is a potential safety concern, and it's always necessary for the members of the program to do their own due diligence. They need to, as you mentioned, if the FDA does not support this, they need to make up their own decision. So our signals is very much in a way suggestions, and a lot of work going into the evaluation, but you need to consider, is this appropriate for my population? So the responsibility for action is always on the national pharmacovigilance and the regulators. And Ying, you can please help me out.
Thank you. Thank you for the question. And I think you have heard Annette's the last presentation, what we focus at the UMC. We usually do not repeat what other agencies already started or already have ongoing signal assessment. And we focus on the global perspective. And if the signal is only from the US or UK, these countries have a capacity to assess. And we did publish some signals and shared in the VGLIS with other national authorities. Some of them have become labeled, some have not. But signal as for the other area of pharmacovigilance is about prioritization. For some signals, it might be followed up urgently in studies, and some signals may take time to prove, to confirm or not confirm. So time will tell. So the focus UMC has focused is considering the clinical relevance and to see if there are possibility to raise awareness for patients and for healthcare professionals to prevent or minimize a potential risk. And so that's the prioritization, one of the prioritization criteria.
Thank you. Anybody else wants to add, or we go to some more concrete questions? No, I don't see. So then I have a question here, which is very concrete. How to map adverse events of special interest, ISIS, to MEDRA? Is it possible for national regulatory authority of a member state to obtain the mapping list?
So the BRITO collaboration has issued some guidance on how to map the adverse events of special interest to MEDRA. So there exists some guidance. And then one of our medical doctors has put a lot of effort into developing this and evaluating these connections to MEDRA. And I would say that if you're interested, let us know, and we can see what we can do when it comes to sharing this information. It would be with a lot of caveats, of course, but it might be possible.
Yes, ACs are mapped to MEDRA where possible to pre-existing SMQs, but sometimes with adaptations or based on pre-existing case definitions, as Helena mentioned, the Brighton criteria. So the ACs definitions we use are only for internal use.
Thank you. And let's go to the next question. How countries develop clustering tools or use UMC tools for the data? So how can countries develop their own clustering tools or use UMC clustering tools for the data? So that's very concrete question.
Yeah, probably for me, I would guess. So in terms of clustering tools specifically, so we have a publication which is currently under review, which I think would be a good place to start. So hopefully that will emerge within the next couple of months. And alternatively, I think what I can do is I can type an answer to this question, right? I can paste a reference in there. So what I'll do is I'll link to another reference where you can read in much more gory detail about how everything works. So if so anybody who wants to implement it, anybody who wants to use it can see that there. In terms of us providing that tool, it's still a relatively young tool. We're still understanding the quirks of it, so we're only using it internally for now. And there's no concrete plans to make it more widely available from within the UMC, but we're publishing stuff about it. So yeah, people will definitely able to try it themselves.
Thank you. There is another question, which is, how should countries with no signal detection software proceed with K-series disproportionality analysis? What denominator values should be taken? So maybe somebody can give at least some answer to this question as well.
So I guess I can answer that one too. So the denominator, or in most measures of disproportionality, of trying to do this kind of case series analysis, assuming that you have a collection of reports on which you want to do disproportionality analysis, the denominator is typically the expected rate of, or the amount of times you expect to see a certain drug event combination or a certain event, with the numerator being the number of times you actually see that event. So I guess the answer is the denominator is, given your data set or given what you can find, you compute the expected number of times that you would see a certain adverse event. I can give, I'll also post a reference to your question in the chat to give some more mathematical detail about specifically what we do at the UMC and specifically what numbers are plugged in there.
Thank you. There is also something very, very concrete question. Aside from the Oracle Argus, what other softwares or platforms are used for pharmacovigilance and signal detection?
Yes, so that's a whole market out there for this kind of tool to both to collect the pharmacovigilance data and also to analyze it. As mentioned, Oracle Argus would be one. I don't have a full list. I know that some systems do use Vigibase data. A couple of more examples would be RS Global and Advera. Those are a couple of more systems that use Vigibase data that you can use that for a background in signal detection in their systems. But I would say perhaps that's a question for the audience. Maybe they use a lot of other systems as well. But no, I don't have the full list, but there is definitely a market for those kind of tools.
Thank you. And then again, something which is quite concrete. Can any suspected, unexpected serious adverse reactions, SUSAR, that have occurred up to date be shared with us attending this webinar? So.
I can take this one. The SUSAS is a serious, unexpected, suspected adverse reactions. And these are usually company-specific terms because it's according to the labeling. And these terms have a special status. They have to be monitored and reported to the regulatory authorities is required. So in short, UMC will not share information about SUSAS, but we do share assessed signals by UMC with the companies who have a concerned product. And UMC assessed signals will be communicated with the company before we put them into Vigilize. But maybe we can sorry. Sure. Sorry. Maybe we can provide custom searches, as Helena's last slide shows. Is that what you want to add, Helena?
No, I was thinking to add also the information that when it comes not to SUSAS maybe, but to signals in general, many of the international regulators during the pandemic implemented transparency measures. So many of the information you can find on the national regulators website if you're interested.
And let's go immediately to another one, which is a very concrete short question. Are there also adverse drug reactions collected about Sinovac? So it's just one vaccine. Are they collected? I would assume yes, but you can add probably.
Yes. It's the short answer.
Yeah, but of course the numbers of reports, individual case safety reports about different vaccines are hugely different. And I think Sinovac is not belonging to the first four, at least, probably not. So probably it's much lower number of reports.
We have less data for Sinovac, but we have data for that vaccine type as well.
So there is, we could say that there is room of improvement about reporting with Sinovac, because according to the number of doses we know delivered and the reporting rate, they are probably not in harmony with, let's say, some other vaccines. So there is improvement clearly possible. Then, of course, there are very general questions, which is, of course, important and interesting, but I assume a bit difficult to answer. How do you implement fast signal detection in pandemic?
Maybe I can start and others can complete. I think in short, it's the preparedness. As Helena mentioned, we start with VGFlow and to receive and handle the reports quickly with a high quality. And then from a data scientist perspective, we have improved the methods and to have a regular frequent signal detection activities. And from a data science, from a pharmacovigilance expert point of view, we are prepared and we do continuous signal detection activities and signal assessment. So I think we prioritize the most relevant signals. The number is not an issue here. There are vaccines exposed very quickly in a very short period to a large number of people. So there are about 50,000 of reports each week. So we need the prioritization and be prepared. UMC has put a lot of resources on signal issues.
Thank you. Then there is a very concrete question, which is how does Vigilize calculate the expected number of reports, even if there is no total number of doses administered reported to the Vigiflow? Can anybody give to this some sort of answer?
I think this is your question, Jim.
Yeah, of course. So yeah, I think I saw there was a couple of questions along these lines in the chat. So so you're right. We don't have the total number of administered doses, which would be the perfect way of calculating it. But it turns out quite a good way of calculating it is just to look at the rest of Vigibase, because Vigibase is so huge. We can look at how often adverse events are occurring when when this drug is not administered from other medications or whatever, and use that to calculate the kind of background expected rate that we should be seeing. That we see adverse events, and then look for a particular specific drug and see if we're seeing them more often. Yeah, I think I hope that answers the question.
Maybe I can add to this that this is a very big caveat when it comes to pharmacovigilance in general. We mustn't confuse reporting rates with incidence rates.
Thank you. And there is a very concrete question to the Uppsala Monitoring Center. Were you able to detect myocarditis with mRNA vaccines and TTS signals with AstraZeneca vaccine from the global reports and using your tools?
The short answer is yes. We could be able to see the disproportionality.
Thank you. So the answer is yes. And can we go then further? There is a very short question. Standardized MedDRA queries. Can standardized MedDRA queries be used in place of clustering algorithms?
Yeah, so that's an excellent question, because I guess on the surface it sounds like clustering is solving the problem which SMQs already cover. But I think what the power of clustering is that it goes into a case series agnostically. It doesn't. The thing with SMQs is they have to be defined ahead of time, whereas clustering can find kind of previously unexpected constellations of symptoms. So that's the short answer. Of course, we use SMQs as well to do signal detection. We're not replacing them. I think that's important to note, is that like all of these tools are supposed to work in tandem, not replace one another.
Thank you. If nobody wants to add, then I go to the next question, which is through the Vigilize, are you able to flag on or propose hypotheses on potential batch-related issues? So if you take like one vaccine, then there are different batches. Is there anything batch-related that has come out from your analysis?
So maybe I can start answering, and Helena, please feel free to chip in. I think the batch-related problems are probably being detected rather on the national level than on the global level, but we do see, and in case we do receive the information, which in the vaccine reports is often the case, that it contains the batch number, for example, we will be able to see, for example, clusters that are locally confined or clusters reporting the same batch. And it is possible to, for the member countries, to extract line listings directly from VigiFlow, from the system that can be used to enter the reports, and which they can use as national database. And these line listings will be very, very helpful to identify batch-related problems.
Thank you. Then there is a very concrete question. What is the recommended minimum number of individual case safety reports needed for signal generation? For example, if a given country would like to generate individually a signal, so it's coming probably from a country, so what would you say to this to answer?
I can start just by saying that
Please.
I guess we're talking about statistical signal detection, because there's no like minimum amount of reports you need to do qualitative signal detection on a case-by-case basis. And then perhaps my colleagues who are more familiar with our work, but I know that we've done some publication on statistical signal detection in small data sets. And I think it turned out that there is really no lower limit. It has more to do with how skewed the data is, if it's a lot of reports in a specific area, or maybe Jim or someone else can clarify that a bit. But I think that piece said 500 reports is okay. That's quite a small database, but you can still have some value from statistical methods in such small data sets as well. Please add.
So we have a really nice publication about this that came out quite recently. So I think that the easiest thing, honestly, is for me. I'll link it in the chat, but I'll just read the conclusion here, which is like a paragraph. To mitigate the risk of highlighting spurious associations with disproportionality analysis, a minimum size of 500 reports is recommended for national databases. For databases or subsets that are not country-specific, our recommendation is 5,000 reports. But I will link that paper. I can recommend it as a nice read.
Thank you, and I think we start to get to the close to our time. There is one question: where is the best place to regularly access COVID-19 vaccine safety analysis data for low and middle income countries? So it's quite a general question, but I think it's important. There is, of course, a certain amount of information available from also WHO headquarters, from well-established regulators, but you may wish to add to this something.
Yeah, so VigiBase, I can just mention that we have, the low middle income countries are well covered in VigiBase. So VigiBase data is, of course, the problem with the public data set that I mentioned in my presentation, the VigiAccess data set, is that you can't really distinguish between the different vaccines in that data set. They're all lumped together as COVID-19 vaccine. And it's also not possible to distinguish between countries. And that's actually a limitation that goes for all publicly available VigiBase data. It's not possible to distinguish on country level, because we need to ensure privacy, so we don't disclose country data, basically. Everything is presented by continent or WHO region in the data sets. But for, as mentioned, the national authorities, they can access Vigilize then. That's free of charge for them to use, and they have access to the full data.
Thank you. And now we have to come to the end because we have exhausted our time. I would like to thank very much all the excellent speakers and panel members for the discussion and the presentations. And I think we couldn't answer all the questions, but we at least tried what we could. So thank you, everybody. Thank you very much.